This protocol is particularly relevant because it allows the detection and selection of single cells from a pool of mixed cells for subsequent downstream analysis at a single cell resolution. The main advantage of this technique is the potential to separate individual circulating tumor cells, or CTCs, from blood cells for a range of analyses that require high purity.
Begin by starting up the Micromanipulator software and switching on the Micromanipulator. Connect the Micromanipulator to the computer and click Connect and Initialize Device to initialize the robotic arm and the microscope stage. Clean the external and internal surfaces of the protective cabinet with ethanol and close the protective cabinet after every manipulation of the machine to be able to maneuver the device through the computer. Install a new 20 to 30 micrometer glass capillary on the robotic arm and flush system oil through the system to remove any bubbles in the tubing. Fill sterilization tank 1 with 70% ethanol, sterilization tank 2 with sterile, nuclease-free water, and the buffer tank with sterile Dulbecco's PBS. Sterilize the capillary 2 times with 70% ethanol and replace sterilization tank 1 with sterilization tank 2 to use the sterilization function to wash the capillary in water at least 3 times. In the Micromanipulator software, start a new experiment and select the type of picking experiment from the automatic and the manual selection modes. Configure the deck tray to specify the positions of the sterilization tank, buffer tank, and depositing tray, and set the temperature of the liquid tanks and the selected depositing tray to 4 degrees Celsius. Position an ultra-low attachment plate containing the released CTC solution under the microscope.